Our last speaker is David Sachs, the co-founder and chief scientific officer, ONL Therapeutics. Thank you, David.
Great. Thank you very much to the organizing committee for allowing me to speak to you today. Can we have the slides up, please? Wonderful. So I'm going to talk to you about ONL Therapeutics. And we are living our values every day in order to achieve our mission, which is to help patients see the future. We are a clinical stage biopharmaceutical company based in Ann Arbor, Michigan. We recently closed a $65 million Series D, and this is to help us advance our goal of bringing these novel neuroprotective therapy to patients. You can see who invested in us, and I'd really like to thank the ophthalmology-focused venture capital groups, many of whom are in the audience, for supporting us through our journey. What do we do at ONL? We're developing zelafacilitide. This was formerly known as ONL 1204. We just got the name approved. We are targeting a novel and differentiated mechanism of action, the upstream activator of cell death and inflammation, the FAS receptor. So as I said, FAS does two things. When activated, it initiates cell death, but it also, at a very high level, but it also is responsible for turning on the inflammatory cascade. By blocking the FAS receptor, we block both arms of the dysfunction, both pathways. Zelafacilitide is a 12 amino acid peptide. It's delivered intravitreally, and let me show you. We're using it in this pipeline. We have a robust GA trial, and I'm going to talk to you specifically about that today. But we also have active programs in glaucoma and other indications. So what does zelafacilitide do in geographic atrophy? In a phase 1 study, we had two components. The first component was, this was the first time it was delivered in this indication, a dose escalation study, 50 micrograms, 100 micrograms, 200 micrograms, 2 patients each, a single injection, patients monitored for 6 months. This was followed by a second component to the study, where we had a natural history treatment phase, natural history phase followed by a treatment phase. We recruited patients. They were monitored for 6 months to get an assessment of their rate of growth of their lesions. And then they were randomized to a low dose, a high dose, or to sham. They were given an injection. 3 months later, a second injection, followed for an additional 3 months. What did we see? Zelafacilitide was safe and well tolerated in these patients. We had 4 serious adverse events, but they were all unrelated to zelafacilitide. In the component 1, we didn't have a control arm, but we did have the fellow eye. With a single injection measured at 6 months, we saw about 42% reduction in the rate of the lesion growth compared to the fellow eye. And look, on the right, potential for some vision gain, as we see in the study eye versus the fellow eye. In the second component, these were patients who were randomized to a low dose, a high dose, or a sham. Here we see the rate of the lesion growth in the 6 months of the treatment phase. The sham grew about 1.8 millimeters squared per year. That's what's to be expected. But there was a dose-dependent reduction up to about 50% with the 200 microgram dose. On the right-hand side, you'll see the vision loss across the whole study. And we saw that their sham patients were losing vision with the potential for vision improvement in the treated patients. Let's ask, does this pass the gut test? Does this make any sense? Is this possible? Remember, I told you that blocking the FAS receptor does 2 things. It blocks cell death. That's represented on the left-hand side of the curve, the left-hand side of the slide. When you block cell death, you block lesion growth. On the right-hand side is the anti-inflammatory effect, or what we posit as such, the improvement in function. Inflamed, sick cells don't see well. You relieve the stress, you relieve the inflammation by blocking the FAS receptor, and there's the potential that they'll actually do what they're supposed to do, which is see letters on an eye chart. So this was very exciting. In our opinion, it passes the gut test, and we're very happy to announce that we're launching now the Galaxy trial. This is a phase 2 study of zelfacilitide in patients with geographic atrophy. This is going to be a low dose, a high dose every 3 months versus a sham. We'll have the high dose also every 6 months against a sham. This is a multinational study. We will have in the United States a reference arm. So in summary, zelfacilitide is a well-differentiated product addressing a unique mechanism of action. We're blocking the FAST receptor to block upstream activators of cell death and inflammation, and emphasis on the 2 pathways and the upstream location of our mechanism of action. And, compelling dosing frequency with the potential to have functional as well as anatomic benefits. We're now enrolling, and I thank you for your attention.